A 2026 paper published in Cell Stem Cell details the exact mechanical pathway through which sleep deprivation physically damages the human gut. Clinicians have long documented a strong correlation between chronic sleep disturbances and the onset of severe gastrointestinal pathologies. The persistent gap in neurogastroenterology has been identifying the physical transmission mechanism. Researchers observed the brain communicating distress to the gut, but the precise biological relay remained undocumented. Previous models pointed to systemic stress hormones like cortisol. However, a diffuse endocrine response fails to account for the highly localized, specific structural damage observed in intestinal tissue following acute sleep loss. This study isolates that damage, focusing squarely on the degradation of intestinal stem cells, or ISCs, which act as the regenerative engine for the entire gut lining. The researchers identified a direct, high-speed neural circuit, proving that the gut's collapse is driven by a hardwired nervous system reflex rather than a slow endocrine drift. Identifying this precise neuromolecular relay defines sleep-induced gut pathology as a mechanically actionable disease with clear therapeutic targets. Here we see the defect's origin, the dorsal motor nucleus of vagus, or DMV, in the brainstem. Sleep deprivation hyperactivates these neurons, forcing an elevated firing rate. This rapid-fire distress signal travels down the vagus nerve, aggressively ejecting acetylcholine into the gut tissue. This is a targeted, aggressive chemical release, entirely distinct from the diffuse wear and tear of general physical exhaustion. This rapid flood of acetylcholine bypasses the standard regulatory checkpoints normally maintained by the local enteric nervous system. Behavioral sleep deprivation effectively weaponizes the vagus nerve, bombarding the delicate gut microenvironment with unregulated excitatory signals. The primary recipients of this aberrant acetylcholine dump are the enterochromaffin cells, embedded directly within the gut lining. Excess acetylcholine binds to muscarinic receptors on the enterochromaffin cell membrane. This forces a massive efflux of serotonin out of the cell while simultaneously suppressing reuptake channels. A chart overlay tracks this toxic concentration, a massive 5-HT spike. The local extracellular environment rapidly fills with an aggressive accumulation of neurotransmitters, transforming a balanced biological space into a saturated chemical pool. This sudden density of serotonin completely overwhelms the immediate tissue homeostasis, breaking down normal intercellular communication. The vagal relay takes the standard signaling molecule and turns it into a concentrated biological hazard, positioned directly adjacent to the gut's most vulnerable stem cell populations. Those vulnerable targets are the intestinal stem cells, which reside at the base of the crypt-villus architecture. Pooled 5-HT molecules exhibit high binding affinity, attaching specifically to HTR4 receptors on the stem cell membranes. This hyperactivation triggers an intracellular cascade of reactive oxygen species, inducing extreme oxidative stress. Consequently, the entire crypt-villus architecture physically shortens and blunts. The once healthy columnar tissue rapidly collapses under the extreme stress, completely losing its strict structural integrity and vital regenerative capacity. Alongside this architectural shrinking, the tissue suffers the targeted apoptosis and total loss of protective Paneth cells within the crypts. The simultaneous reduction in villus height and the eradication of Paneth cells critically compromises the entire intestinal epithelial barrier. Through this precise molecular chain, the brain-gut relay forces the physical exhaustion and complete structural collapse of the gut's regenerative engine. This finding refutes the reliance on generalized systemic stress models in neurogastroenterology, proving a direct neuromechanism for the damage. By identifying localized molecular receptors, clinicians now have targetable points of intervention for the physiological fallout of sleep deprivation. The first potential pharmacological intervention lies upstream, modulating the vagal acetylcholine output at its source to prevent the initial neural trigger. A localized strategy targets the gut directly. This diagram demonstrates a pharmacological antagonist blocking the stem cell's HTR4 receptor, deflecting excess serotonin. This decouples the toxic 5-HT spike, maintaining tissue viability and regenerative capacity, even during acute sleep deprivation. These specific therapeutic strategies offer immediate applications for high-stress populations facing acute sleep loss, such as shift workers or intensive care patients. Precise neuromolecular targeting can chemically sever the link between behavioral sleep loss and physical gastrointestinal damage. A primary limitation of the current study is the need to clinically map the differences between acute sleep deprivation and chronic long-term insomnia models to see if the mechanism adapts over time. There is also a critical open question regarding how this hyper-localized serotonin spike might disrupt or interact with the broader ecology of the gut microbiome. To summarize the pathology, the top-down circuit initiates with the hyperactivation of the dorsal motor nucleus of vagus in the brainstem. That signal propagates downward, forcing a rapid vagal acetylcholine flood, which triggers a dual-pronged serotonin spike from the enterochromaffin cells. The elevated serotonin binds to HTR4 receptors, inducing the severe oxidative stress that degrades the intestinal stem cell niche. For precise molecular protocols, extended datasets, and a comprehensive breakdown of the methodology, clinical researchers should refer directly to the primary literature. Cognitive and behavioral states physically dictate stem cell survival through hardwired neural circuitry, providing a strict mechanical basis for the brain-gut axis.